This conference will now be recorded.
Hello, and welcome to the Invasive Mussel Collaborative webinar, Outcomes and Lessons Learned from Recent Zebra and Quagga Mussel Control Studies. This is Ceci Weibert with the Great Lakes Commission. On behalf of the Invasive Mussel Collaborative, thank you for participating in today's webinar. The Invasive Mussel Collaborative was convened in 2015 to identify opportunities to advance scientifically sound technology for management and control of invasive dry-seeded mussels to produce measurable ecological and economic benefits. If you would like to know more about the Invasive Mussel Collaborative, we encourage you to visit our website. Today's webinar is part of an ongoing series hosted by the Collaborative. The purpose of these webinars is to facilitate learning and information sharing on topics of common interest within the invasive mussel community. It will be recorded and archived along with past and future webinars on our website. This webinar will cover 3 recent zebra and quagga mussel control studies using Zequanox on Round Lake in the Upper Peninsula of Michigan, Zequanox in Lake Michigan's Good Harbor Bay, and EarthTec QZ on Lake Minnetonka in Minnesota. We are pleased to have speakers from each project joining us today. While we always strive to minimize technological issues during these webinars, we apologize for any issues that may arise. Further, I'd like to cover a few logistical items before we get to the presentations. Audio for this webinar is automatically streamed through your computer speakers once you join the GoToMeeting event. You also have the option to dial into the webinar using a telephone. To change your audio to a phone line, select the settings icon in the top right-hand corner of the webinar screen and click the phone tab. A call-in number will be displayed as well as the access code and audio pin to join. Once you are connected from your phone, click the red Turn Off Computer Audio button to reduce interference. In order to minimize the potential for interruptions and background noise, we have automatically muted all attendees. Attendees will remain muted throughout the webinar. That being said, we do encourage and will accept written questions from attendees during the webinar. If you have a question, you may type it into the chat box signified by the chat bubble icon in the top right-hand corner of the webinar screen. If you are experiencing technical difficulties, please send a message through the chat box as well. Questions may be submitted at any time during the webinar. Depending on time, we may answer 1 or 2 questions following each presentation, but the majority of questions will be addressed after all presentations have been given. We will do our best to take all questions or comments during the webinar. If you need to leave the webinar before we have gotten to your question, we encourage you to review the recording that will be posted on our website to hear your question and answer. Once a presenter is sharing their screen, a banner with sizing and screenshot zoom features will pop up on the right hand of the shared screen. You may increase the size of the shared screen by either selecting the detached camera buttons that will open the screen in its own window, or by using the zoom buttons. Our first presenters this morning are Diane Waller and Angelique Dahlberg. Diane Waller is a research fishery biologist with the USGS Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin. Her research focuses on development and evaluation of selective management strategies for aquatic invasive species, particularly zebra and quagga mussels. She is particularly interested in developing control tools and application methods that minimize impacts to non-target species. Diane also conducts research on native freshwater mussels, including life history studies, development of health assessment tools, and investigating causes of mussel mortality events. Angelique Dahlberg is a graduate student in the Phelps Lab at the Minnesota Aquatic Invasive Species Research Center, working towards a PhD in conservation sciences. She received a master's in integrated biosciences from the University of Minnesota Duluth and a bachelor's in biological aspects of conservation from the University of Wisconsin-Madison. Angelique also spent 5 years working for regional nonprofits to manage and mitigate the impacts of invasive species. Angelique's current research investigates suppression methods for controlling zebra mussel populations in Minnesota lakes. And so with that, I think that Diane and Angelique, if you have presenter status, you can go ahead and start sharing your screens.
Okay, thank you for that introduction, Ceci, and welcome to all the attendees out there. I'm Diane Waller, and I'm going to be tag teaming this presentation with Angelique. So I'll give her just a moment to get the screen up.
Yep, this is Angelique. I actually do not have presenter status right now. So if someone who does have that could assign that, please. Thank you.
So thank you again for the invitation to present this collaborative research between USGS and the Minnesota Aquatic Invasive Species Research Center on an investigation to use low-dose copper for zebra mussel suppression. I was involved in the early planning stages of this work in 2017 and 18, but Jim Loma was the project manager until early last year and really spearheaded the study. So Jim and his crew, with Angelique's assistance, executed the treatment and sampling. I've taken over the long-term monitoring of the project that we're doing this year and next year, and then a second phase of the work that will also start next year. Next slide. Funding for the project came from the Minnesota Environment and Natural Resources Trust Fund and the Minnesota Aquatic Invasive Species Research Center, along with funding from Hennepin County, Pelican Lake Association, Fletcher Family Foundation, and some others shown here. And I have to mention Tonka Bay Marina was really invaluable in providing support for technical operations and really study setup on site. And a number of local and state partners provided help with outreach, permitting, and logistics. Next slide. I'll start with a brief overview of copper use in lake management and then describe our project objectives and the study design. And then Angelique will take over to present the results of the 2019 treatment and our plans for future research. This presentation is not an endorsement of EarthTec products. Next slide. Copper has a long history of use in aquatic systems, as I'm sure many of you are aware. So copper plating was used to prevent biofouling, and there's many copper coatings still in use today to prevent attachment of organisms like zebra mussels, but it's also been used to control algae, aquatic vegetation, and snails, among other things. In Minnesota, you can see a table on the right-hand side here of the use of a copper in some rapid response or zebra mussel eradication efforts between 2011 and 2019. So a number of them. Some of these treatments also included adding other toxicants with the copper, such as potash and Zequanox. All of these were enclosed partial lake treatments. Copper has also been used in an eradication effort in Nebraska on Offutt Air Force Base. The results here were kind of mixed, as it also killed mussels. It killed mussels, but also caused some significant fish mortality. Treatment of a 12-hectare lake in Pennsylvania in 2017 with the copper product EarthTec QZ reportedly eradicated all mussels from the quarry, and some long-term monitoring will indicate whether or not that actually is the case. So these are eradication treatments that were intended to kill all life stages and used up to 1 milligram per liter of copper, which is the maximum allowed on the label. But the early stage, especially the veliger or larval stage, should be more sensitive than the adult. And studies conducted by Mike McCartney and Renata Clotti compared veliger and adult sensitivity to copper, and they reported concentrations that would produce 50% mortality, or the LC50, for veligers that were about 50 times lower than that for adults. In fact, Claudia reported an LC50 of 19 micrograms per liter of copper for villagers, compared to that's reported for adults of 1 milligram per liter. Next slide. So EarthTec QZ is a copper-based product that's registered for open water use as a molluscicide. These previous applications of copper indicate that it can work, and the research by McCartney and Claudia showed that these lower concentrations, below 1 milligram per liter, can kill villagers. So what other questions do we have? Well, we know that copper is toxic to non-target species. So before copper is used in a Dreissena management program, we really need to determine how to minimize ecological risk, economic costs, and then maximize the long-term benefits to a water body if it's going to be used. So the objectives of this project were to evaluate the effectiveness of a low dose of copper to suppress the villagers and then reduce juvenile settlement after the application. And secondly, to measure the response of non-target organisms to the treatment. And lastly, to see if reducing recruitment 1 year would translate to decreased zebra mussel density the next year and a benefit to native communities. Next slide. So a little bit of information on the formulation of copper that we tested. This is EarthTec QZ. It's certified for use in potable water supplies. It's a liquid formulation of ionic copper. There is no post-application reentry, water contact, or water use restriction. The label allows treatment up to 1 milligram per liter of copper, and the target concentration for this treatment was about 6% of that, or 60 micrograms per liter. The application was scheduled around peak villager abundance, which occurs around mid-July in Minnesota. So the product was applied only to the epilimnium, so that was the upper 5 to 6 meters. And before each application, the thermocline was measured, and then the volume of water in the thermocline was estimated for the application. The treatments were applied every other day for 10 days, beginning July 22nd through the July 30th. And in addition to measuring zebra mussel response, we also assessed impacts to 4 fish species, a native mussel, zooplankton and benthic invertebrates, and water quality parameters. Next slide. The study was conducted on Lake Minnetonka, which is a sizable lake with a lot of bays in the Twin Cities area. Zebra mussels were first found in the lake in 2010, and they're well established in various areas across the lake. So when control and treatment sites were identified, it was important that the bays were isolated so there was no mixing of the treatment into the control site, but also as similar as possible in water quality, flow, and zebra mussel population. So you can see by the diagram here, there were some differences in the size and bathymetry between the bays. Robinson's Bay was the control site, 37 hectares, a maximum depth of about 19 meters. And the substrate here is kind of sand to gravel. Whereas in the treatment site in St. Albans, it's shallower, a larger surface area, and had really kind of an organic, rich, silty type of substrate. Next slide. 5 locations were identified as sampling sites throughout each bay and then marked with a buoy. And at each of those buoys, test animals were held during the treatment, and plankton toes, benthic grabs, and water samples were taken before, during, and after the treatment. Next slide. So this slide, the graph shows the copper concentration that was measured over that 10-day application period. The dashed blue line is the target concentration of 60 micrograms per liter. But you'll notice the star is the initial application was targeted at 100 micrograms per liter, just because we knew the concentration would drop. And then the plus signs are those bump treatments on alternate days to maintain the concentration above the target. The dashed black line is the copper measurement that was made in the field with a Hawk meter. These were used to determine how much to apply on those bump treatment days. The black solid line are samples that were returned and analyzed on the ICP in the lab. So you'll notice that's more accurate reading, that the daily mean for the ICP readings was about 15 micrograms higher than the field reading. But overall, the treatment did maintain the concentration above target over the 10 days. What's not shown here is that within 2 weeks after treatment, copper concentration had decreased 50% and was back to background between 60 to 90 days after the treatment. And I'll turn it over now to Angelique to present results.
Thanks, Diane. All right, I will take it from here. So to understand how this treatment impacted zebra mussels, which were our target organism, we monitored veliger densities before and after treatment in both bays, juvenile presence on settlement plates in both bays, and presence of living adults in the resident populations of both bays. Preliminary analyses of our data indicate treatments effectively reduce zebra mussel veliger density. So here you can see that pre-treatment veliger density in both bays was similar and even slightly higher in our test bay. And after 14 days, the density was around the same level in the control bay, but veligers were nearly absent from the test bay. Additionally, the treatment effectively reduced zebra mussel settlement. We placed settlement plates in both bays, retrieved half of those 30 days post-treatment, and then retrieved the other half at 90 days post-treatment. At both time periods, at both the 30-day and the 90-day, we saw very few attached mussels in the test bay, while the numbers we saw in the control bay were more than 3 orders of magnitude greater. So this indicates strong treatment-related density reductions. Finally, we also observed a decrease in the number of live adult resident zebra mussels in the treated bay. To assess this, we had a scuba diver run transects in both bays in early July before the treatment, and then again in September, nearly 2 months after the treatment. The scuba diver enumerated living and dead resident mussels in quadrats along the transects. The figure on the left in this screen shows live adult zebra mussel density, and the figure on the right shows the percentage of live adult zebra mussels. So that takes into account the dead mussels. Prior to treatment, we saw similar densities in both bays of living adult mussels. And after treatment, we continued to see high numbers of living adult zebra mussels in the control bay, but density and percentage alive in the treated bay declined by 2 orders of magnitude. And so these were our findings on impacts to resident zebra mussels. I'll also talk later about the survival of caged adult zebra mussels that were held near the sampling buoys and that were also reduced in the treated bay. In addition to impacts to zebra mussels, we also looked at a variety of non-target impacts. First among those, we monitored water quality and chemistry metrics, 2 of which are displayed here. We observed a slight depression in dissolved oxygen concentrations in the treated bay after exposure. However, the dissolved oxygen did not drop to levels that would cause harm to aquatic life. And other water quality and chemistry metrics remained similar throughout the study period. We found some impacts to algal productivity. We collected chlorophyll A samples and took Secchi disc depth measurements 1 day prior to initial treatment, 1 day after the first day of treatment, and 14 days after treatment ended. And in the treated bay, both chlorophyll A concentrations and Secchi disc depth measurements indicated a substantial treatment-related increase in phytoplankton immediately after treatment, followed by a decrease 14 days after treatment. Meanwhile, in the control bay, we saw an increasing trend in phytoplankton community, seeing both an increasing chlorophyll A concentrations and decreasing Secchi disc depth measurements. We found a decrease in the abundance of both benthic invertebrates and zooplankton. For both organisms, we collected samples 1 day before treatment, 1 day after treatment began, and then 14 days after treatment ended. Benthic inverts were collected in ponar grab samples, and zooplankton were collected using plankton toes. For benthic invertebrates, within each bay, there was no statistical detectable difference between the sampling periods, although we did see general trends. For those trends, we saw an increase in abundance in the control bay and a decrease in abundance in the test bay. For zooplankton, in the control bay, zooplankton density was greater 14 days after treatment compared to pre and 1 day after treatment measures. There was a considerable reduction in the density of native zooplankton in the treated bay, both immediately after treatment and at the start of recovery 14 days after treatment. And we're also doing some community composition analysis on these numbers. In addition to those organisms we monitored that were already living in the lake, we also monitored some caged organisms, 4 fish species, a native mussel species, and then adult zebra mussels. We placed fat bucket mussels, fathead minnows, bluegills, largemouth bass, yellow perch, and adult zebra mussels in independent cages beneath each buoy in both bays. The cages went in the day before treatment and were retrieved the day after treatment ended. We calculated survival based on the number of stocked organisms and not the number that we were able to recover. This was because we had some challenges arise with the recovery of our fish. For example, mortalities might have occurred early, tissues may have degraded or been consumed by other fish in the enclosure. So this limited our ability to accurately determine mortality without making some assumptions. And so we based our calculations on the number of fish originally stocked. The cage was the sample unit here, and the sample sizes for each bay were either 4 or 5. There was some handling stress during stocking and additional stressors at the buoy locations, either from depth, temperature. There were definitely predators that were present. There was an active boating community, including wake boats. So it certainly was not an area without stress to these animals. We saw that yellow perch and largemouth bass had similarly poor survival in both bays, maybe due to some of that handling and holding stress. But a main takeaway from this work is that treatment-related mortality was most apparent for zebra mussels and for fathead minnows. And one thing to consider as we look at this, survival of adult zebra mussels was greater than 60%, but this was only 1 day after the treatment that we removed them. And based on our scuba survey of the live resident zebra mussels in the bay, we suspect that there was some delayed mortality happening. So once those fish and mussels were taken out of the lake, we sent them to the lab and analyzed their tissues for copper accumulation. In the treated bay, we saw that zebra mussels accumulated the most copper in their soft tissues, followed by native mussels. Fathead minnows in the treated bay had tissue copper concentrations that were an order of magnitude higher than those in the control bay and appreciably higher than those of other fish species. So we did see some trends there. One thing to recall as we look at this data, though, is that the sample size is less than or equal to 5 for all of these. So that leaves a little bit of room for us moving forward to continue looking for answers to some of these questions. In summary, though, treatments effectively reduce zebra mussel veliger density, juvenile zebra mussel recruitment, and live zebra mussel density in quadrat samples. Non-target impacts varied. Relative zooplankton mean density was reduced at immediately after treatment and showed some recovery at 2 weeks out. Chlorophyll A concentration increased after treatment, and survival and copper residue in fathead minnows suggest a sensitivity in that species. So we do have a few next steps moving forward. We want to learn what the long-term responses of zebra mussels and non-targets are to copper and get more information on that to determine if we can effectively apply less copper. So we are continuing monitoring in Lake Minnetonka this year and next year, and would like to add a second lake to our study to add some replication to this and continue working toward these answers. And with that, I will conclude our presentation, and we can take some questions, I think, at the end. So thank you.
Thanks, Angelique and Diane. We did get one question, but we're going to hold it to the end, and we are going to continue on now. So let that be a reminder if you have questions for Angelique, Diane, or any other of our presenters today to please enter those into the chat window. So moving on, our next presenter is Caroline Keeson. Caroline is the Monitoring Programs Coordinator for Tip of the Mitt Watershed Council and is responsible for all field work, including water quality monitoring, shoreline surveys, and aquatic plant surveys. She also manages 3 volunteer programs totaling over 75 volunteers. She has a Bachelor of Science from Michigan State University and has spent 9 years working in water quality resources in Northern Michigan. So thank you, Caroline, and the floor is yours.
Hi, can you see my presentation okay?
Yes, we can.
Okay, good deal. All right, so I'm going to present on an application of a biocontrol called Zequanox, which should control zebra and quagga mussels on Round Lake. So just a little bit about the Tip of the Mitt Watershed Council. We're located in northern Michigan, and our mission is to protect lake streams, wetlands, and groundwater. And we do that through research, water quality monitoring, restoration, policy, and education. And our service area, so this is northern Michigan. We have a 4-county service area and the watersheds contributing to those areas. And we're going to be talking about, I don't know if you can see my mouse, but this little area in between Harbor Springs and Petoskey in Emmett County. So just a little bit about the project. We had quite a few partners, the USGS, Michigan Natural Features Inventory, many different researchers from the University of Michigan Biological Station, Marone Bio Innovations actually supplied the Zequanox, and then of course us. So I'm going to talk about the preliminary surveys, treatment plot selection, how we applied the Zequanox, some of the surveys that we did before the treatment, what the actual application looked like, and then post-treatment monitoring and evaluation. So a little bit about Zequanox. Since the early 1990s, people have been trying to find a biocontrol for zebra and quagga mussels. And I think it was in, let's see, 2007, Marone Bio was able to license or get a license for something called Pseudomonas fluorescens, which is a soil bacteria. So it's a bacteria that they discovered destroys the digestive system of zebra and quagga mussels. And so they got a license to use that, or it was licensed specifically to Marone Bio, and then they actually got a EPA approval for use in enclosed areas in 2012. And then in 2014, they got an approval for an open water application. So previously to this study, I think there were 5 or 6 or even more studies where they used Zequanox to control zebra and quagga mussels, but they used semi-permeable barriers to keep the concentration really high. That's because this is applied in in the water column. But the thought was that those barriers were actually a prohibitive cost, and they, you couldn't apply as much Zequanox because it was more expensive, there was more labor involved, they were harder to use. And so really this study tried to see, if Zequanox could be useful for zebra and quagga mussel control on an inland lake, and also could they get rid of that extra cost and just start applying it similar to other aquatic herbicides or pesticides, for different invasive species. So we started by searching for some suitable lakes, and the requirements were if they had to have zebra or quagga mussels, and they had to have the unionid mussels. So we were looking at Crooked Lake and Pickerel Lake. Those were some options. Here's somebody checking out these lakes, looking to see if they have what they need. We actually ended up settling on Round Lake. So Little Traverse Bay is part of Lake Michigan, and then Round Lake is right between Harbor Springs and Petoskey. And you can see here's a larger reference map. The UP is actually north of us. We're in the lower peninsula, not the upper, but we're very close to the bridge. So Round Lake was chosen because the infestation was more recent. It was a less aggressive infestation. The surface area of that lake is about 350 acres. The shoreline is a little over 3 miles, and then the maximum depth is only 12 feet. So it was a pretty small system to get around in terms of logistics and setting up our treatment plots. Here's what the lake looks like. This is just a little curve of the lake here, but it's pretty small. You can definitely see right across it, no problem. So the treatment plots, we placed 4 control plots and 4 treatment plots for 8 total. Each one was 3/4 of an acre in size, and then the total treatment area ended up being 3 acres. And so this is a map. The control plots are, I believe, in the yellow, and the control areas are in blue. The treatment areas are in the beige color. And so they were done in pretty shallow areas. You can see mostly, it was easy for divers to get down and get the mussels, and that's where they were going to live anyway, in those shallower areas. So here's another map of that. I just wanted to show that we picked plots that were near public land, we weren't disturbing any residences. So this northern area is owned by a local conservancy. They owned this land down here in the southern area and to the east here. So they were also a good partner in helping us decide some of our sites based on land use. So how was this Zequanox applied? Well, it was applied from a boat. They had a mixer on the boat, and there was a hose system underneath the boat. It was actually 12 hoses, I believe. And this was developed at the University of Wisconsin. And the idea was that the Zequanox would reach optimal concentration at the bottom 0.7 meters of the water column. So we were trying to keep the Zequanox really close to the bottom. Here's what the boat looked like. I think you saw this in another slide. And it actually went in reverse to make sure that the boat motor didn't screw up the concentration of the Zequanox. It was applying Zequanox at 0.6 meters deep and at 87 liters per minute. So they actually applied this in a back and forth manner over these treatment plots, and they used a GPS to make sure that they were on track. So we got a pretty accurate application pattern. And I believe these are photos from Marone Bio, but this is what it would look like. It's like basically forms a big cloud of Zequanox in the lake. So what actually happened? Well, we applied Zequanox in the morning during 4 different days, and it actually rapidly dissipated. So the concentrations, we collected concentrations 1 hour and 8 hours later, and they were very similar. So the thought is that it dissipated too quickly to actually get a sufficient concentration to kill zebra mussels. So looking at how we evaluated the effectiveness, We quantified zebra mussels before and after. We took containment samples, and we used quarter-meter quadrats throughout the plots. So I forget how many quadrats we had in each plot, but each one we used it to measure. We collected mussels, native mussels, and zebra and quagga, and we did a bunch of analyses on them. These little boxes here actually also had zebra mussels in them, and that's what they would do some of the tissue analysis on. So some of the mussels were actually tagged, and they did a glycogen analysis. This was done by the USGS. So the glycogen is a way to measure the energy of the mussel, and so we use that to see if their energy levels were different before or after treatment. We would expect them, if they were being affected by the Zequanox, we would expect the energy stores to be lower post-treatment compared to pre-treatment. So a little bit about how this ended up. I just wanted to show, here's how the treatment plot was actually split up. There's 3 colors on this square, so they actually sampled the core of the treatment plot, the fringe, and the edge, and I believe all the samples there weren't really any differences between those areas. So the mean mortality of the caged zebra mussels, so those were those little colored boxes, it was actually higher than the control plots, but they couldn't detect what differences were caused by the treatment. They did expect that the soft substrates may have resulted in poor habitat, and that may have contributed more to mortality than the Zequanox application, because the mortality was found to be lower in the plots with firmer substrates, so it was easier for the zebra mussels to attach and stay living there. They also did find that the glycogen, so those are the energy stores, that they were decreased, I believe, in both the zebra mussels and the native mussels, but they actually attributed that to differences in food resources, handling, relocation, and habitat changes, because they had to assume that because they had tested the Zequanox concentrations, they knew it would be too low to actually affect any of the mussels. So they had some different reasonings as to why their energy stores depleted. So we also did an environmental response assessment. So this is what a lot of our partners actually took on. So the biostation took on a lot of the physiochemical monitoring, and then the USGS did some of the other analyses. So we tested for algae, invertebrates, emerging aquatic insects, and then phytoplankton species. And really, we found no differences in really any of these parameters. So between pre and post treatment, again, we monitored before, 1 day after, 7 days, and 1 year after. There was really no change in the phytoplankton, no measurable effect on emergent insects. And this is actually a graphic of the emergent insects differences. And because all the arrows are kind of facing different directions, we can't really say that there was a significant shift in insects or populations from pre to post application. So there were no changes to total phosphorus, total nitrogen, and CBOD, no changes for physiochemical measurements. The only thing they noticed was a slightly higher chlorophyll A in the sediment samples, and then higher turbidity, which was probably right after the application. And if you see those pictures, you understand why, knowing that Round Lake is has a lot of soft sediments. It would make sense of why that application would stir things up for a little bit. So to measure some of the mussels, we actually had divers go down and get the mussels, whatever mussels they could get out of these quarter-meter quadrats. They put them into little mesh laundry bags, actually, that one of our mussel experts ended up buying at a grocery store. And it was pretty murky down there. And so they would actually pick up the mussels from the lake bottom, transfer them to a couple people who were in kayaks, and then the people in kayaks would come back to the boat, which is this picture on the bottom left. And so then they would give the mussels to the people on the boat, who would identify what kind of mussel they were, and then they would measure with calipers the shell length and the shell width and identify whether they were male or female as well. And so here's all the these little yellow dots are all the little quadrats that we had set up in these plots. So what did we find with the native mussels? We found fat muckets, giant floaters, and eastern pond mussels. I think most of the eastern pond mussels were actually dead. So we could only use the fat mucket in large enough quantities for a paired t-test, and we found no significant effect on the fat mucket number or the mean shell dimensions. There was also no significant effect on attached zebra mussels either, because we would count whether or not they had live or dead zebra mussels on each mussel. So there was also an education outreach component to this. We have what we call our Watershed Academy, and it's a school-aged program for high school students. We have a education coordinator who goes in and trains kind of a subset of students at a bunch of area schools on why water quality is important and how to monitor it. And they also have a field day. And so he added to his program some lectures on zebra mussels and quagga mussels and the project. And then they also went out into the field and did some mussel sampling. So the lessons learned from this, because we really couldn't see an effect from the Zequanox after application, is that it really does require some kind of containment. The other idea from this project is if there was a better formulation that reduced exposure time or altered the delivery method, then the Zequanox would stay in the 0.75 meters of the surface. It would stay in that area more. And so the idea was possibly a granular or similar slow release Zequanox tablet would work better than just the liquid application. And then in terms of the education outreach, the in-school presentations really worked better than the field work. And I'm not really sure why, but I would guess, it's a large group, working on some very small things that are hard to find can be difficult to coordinate out in the field. So that's pretty much all I have. I think we are holding questions till the end. Is that right?
Yes.
Okay.
All right. Thank you, Caroline. And our final presenters for today are Erica Jensen and Doug Bradley. Erica Jensen joined the Great Lakes Commission staff in 2006 and is the Great Lakes Commission's Aquatic Invasive Species Program Manager. She serves as coordinator for the Great Lakes Panel on Aquatic Nuisance Species, Invasive Mussel Collaborative, and Great Lakes Phragmites Collaborative, and provides strategic, administrative, and programmatic support. She also works with a variety of partners while managing other AIS initiatives within the GLC, and she has a master's degree in environmental management from Duke University and a bachelor's degree from Michigan State University. Doug Bradley is a senior project scientist at LimnoTech in Ann Arbor, Michigan. He has over 25 years of experience as an aquatic biologist and is the lead scientist of LimnoTech's biological services. Doug served as the project manager and technical lead for this Zequanox pilot study. And you look like your screen is up, Erica, and you're good to go.
Great, thank you. How's my audio?
It's perfect.
Great. So I just wanted to make a few quick remarks about the Mussel Collaborative and the initiation of this project, and then hand it off to Doug to talk about the design and implementation. So as you heard at the beginning of this webinar, which is hosted by the Invasive Mussel Collaborative, we were established in 2015, and we have been working since that time to identify and advance management and science priorities to support development of zebra and quagga mussel control and management tools. We have a multi-jurisdictional, multi-agency and organization membership. And within that membership, we have the 4 founding agencies, the core team, which consists of the U.S. Geological Survey, the Great Lakes Commission, the Great Lakes Fishery Commission, and the NOAA Great Lakes Environmental Research Laboratory. And then within that, we have a steering committee of the collaborative that helps guide collaborative activities and establish work groups to address priority activities of the collaborative. And supporting all of that is the Great Lakes Commission, who provides coordination and backbone support to the collaborative with funding from the Great Lakes Restoration Initiative through an agreement with the U.S. Geological Survey. So a couple of years ago, the Mussel Collaborative, after we had been established, we worked to set up an overarching strategy for goals and objectives that we wanted to address and advance progress on through the work of the collaborative. So in 2018, the collaborative released its strategy to advance management of invasive zebra and quagga mussels with the intent to drive investments in policies and research that lead to outcomes that will help us reduce invasive mussels and their impacts in the Great Lakes region and in other regions of the US and Canada where these mussels are also a problem. We wanted to be able to inform, coordinate, and prioritize work that was already ongoing, recognizing that we still want to be able to allow flexibility in how managers and researchers approach addressing these priorities and working towards implementation. That strategy is organized around 5 goals that represent different management goals for undertaking control and management of zebra and quagga mussels. And the one I want to highlight for purposes of this project is the Mussel Collaborative, primarily representing the Great Lakes region, was very interested in opportunities to advance work that would help us protect and restore critical habitats with ecological value in Great Lakes coastal areas. So we recognize that there are coastal reefs and other coastal resources across the Great Lakes Basin that are impacted by these zebra and quagga mussels. And we wanted to drive investment in research around understanding if mussel control could feasibly be applied in these areas at a local scale to generate larger benefit for the Great Lakes system. Aligning with the release of that strategy, the Mussel Collaborative had an opportunity to fund a demonstration project. And with that strong interest in the coastal work and several of our Mussel Collaborative partners through the National Park Service had already begun looking at Dreissenian mussel impacts in a coastal area of the Great Lakes and had initiated some manual removal efforts. They were interested in expanding that work that was already going on to further investigate the role mussels are playing in that ecosystem, but also pilot other control methods and compare their feasibility to the manual removal efforts that they had already conducted in that coastal reef. So the location of the project we're discussing today is Good Harbor Bay, which is adjacent to the Sleeping Bear Dunes National Lakeshore in northern Lower Michigan on Lake Michigan. The closest marina is Leland, and I believe Doug will have a couple of maps to orient you. But in developing this project, the Mussel Collaborative members settled on wanting to do a demonstration of the Zequanox control method that's already been discussed today, but to do a novel application of that method, both in terms of its location on a coastal reef in Lake Michigan and in the application design in terms of seeing if we could apply the treatment method under a containment barrier to minimize the amount of product that would be needed by delivering it and holding it at the bottom on top of the reef, closer to where the mussels were, closer to where the mussels are. And then along with that, do the associated monitoring of effectiveness of the treatment as well as associated ecological effects. So with that, I'm going to hand it over to Doug to talk about the design and implementation.
Right, thank you. I assume folks can hear me.
Yep, you're coming in clearly.
Okay, and am I sharing the right screen?
We see your meeting invite right now in Outlook.
Dang it. All right, I swear I did this earlier. Okay. How's
There
that?
you go. Mm-hmm.
Okay, and then I just need to dink and dink.
If you go up into display settings, you gotta swap your views.
Display settings.
On the top of your PowerPoint screen there.
Oh, here.
We're seeing your presenter view right now.
Okay, how about I do this? How about I exit out of this? Why can't I do this? That's still not working.
No, if you at the top of the screen where it says show taskbar, display settings, and end slideshow, if you click on display settings, you'll have the option to swap. Yep. There you go.
Okay. Thank you. Sorry about that. Okay. Thanks everyone for the opportunity here. Obviously I'm technically challenged, but we did a really nice job of implementing this study. So that's what I'm going to talk about today. As Erica mentioned, this is a pilot study conducted in Good Harbor Reef, testing the effectiveness of mainly a new delivery method for Zequanox at reef location in Good Harbor Bay, which is, I'll talk about where that is in just a second. Our goals for the project were to evaluate the feasibility and effectiveness of the application methods and see how the benthic community responded to our application methods. We had a heck of a large team because this is a pretty complex project. It required a lot of input from a lot of experienced folks. It required permitting and a lot of implementation challenges that really benefited from this large group. So I can't thank all of these folks enough for helping us make this a successful project, but I won't identify everyone because there's so many. But we really appreciated all the help. And it came together nicely because of everyone's efforts. As mentioned, our study area was off of Sleeping Bear Dunes, south of Leland. Good Harbor Bay is a bay, and it's the reef itself, Good Harbor Reef, is a many-acre reef that's actually within the bay. It's a rocky substrated area, one of the many natural reefs throughout the Great Lakes regions. This one's really good size, but it is covered with quagga mussels. So it was a nice treatment site. Primarily, we knew about it because of all of the work that our project partners, many of the partners have been doing over the years at that location. So we benefited from the experience of a lot of the other previous researchers. Here's a photo of the reef location. As I mentioned, it's been well studied and documented. Other parts of the reef the reef itself is about 30 feet deep. And as I mentioned, it's densely covered with quagga mussels, as you can see in this picture here. Carolyn did a nice job of introducing the Zequanox product, so I won't spend a lot of time on this. We originally looked at several different types of treatment methods, including Zequanox. And given the complexity of just this one method, we chose to really focus our efforts on the Zequanox application. And as was mentioned also, we were trying to reduce the volume of product that needed to be used for treatment. So we really looked at different methods for keeping that volume of this pesticide down. And we landed on what I'm about to talk about. Before I go into that though, a little bit more about Zequanox. It's reactive. Its reaction time and its potency is actually within hours once it's mixed with water. So we really needed to come up with something that a way that would allow us to travel out to the site, set up, run our tests in a matter of hours, and be able to have this concentration that we're targeting that I'll talk about work effectively with our other site constraints. So our bigger question was, can this product be applied in an offshore location using a smaller volume and a controlled and contained approach? As an important part of the team collaboration, given that we're delivering a pesticide into Lake Michigan, we needed to go through a number of regulatory steps. And this is really another part where place where our partners really helped out in getting the regulatory documents together. And I really want to thank also Eagle and the Corps of Engineers folks for helping make the process smoothly through their communications back and forth quickly. We actually received our permit the day before we started implementing. We were actually up there staged and waiting, and we received our permit that in that tight of a time frame. So thank you, Eagle and the Corps. The way we opted, or our approach was to lay out plots. We had a 10 by 10 treatment plots as part of the agreed upon sampling test sample sites scheme. But what we needed to do is figure out a way to develop something that was controllable. So we opted to take these 10 by 10 plots and then turn them into quadrats, and the quadrats are 5 by 5 meters. They're 1 meter tall, 5 by 5 meters in width and height. And this graphic here shows our original planned treatment scheme based on which quadrat we would treat on which days. So we had this numbering scheme and a daily treatment scheme that's depicted on here. The photo there simply shows the markers that our divers used to mark the corner of the treatment plots as well as the quadrats themselves. So what we ended up doing for our containment area design was we worked with our diving partners, Underwater Construction Corporation, and we developed PVC frames, which you see in the picture here. And it's about 1 meter tall frame and then 5 by 5 meter in size. And we worked with the divers quite closely to develop this frame that could be disassembled, transported, and reassembled underwater and easily moved to fit those quadrat plots that we showed earlier, that I showed on the previous slide. We used a heavy-duty tarp to basically cover the area, and then we used sandbags to secure the site to make sure that there wasn't any movement, but also, as importantly, make sure we weren't getting any leakage, so we understood and were able to control how much concentration and volume was introduced into these plots. The upper center photo there shows basically a line that was our delivery system going into the down from the boat to the divers into the containment areas themselves. One of the many tricky parts of this project was being able to get permits to transport liquids out to the site. So we opted to do the mixing and delivery system, develop a mixing delivery system on the boat that would allow us to do all of that on site. So the product, Zequanox, didn't start breaking down prematurely before it was actually delivered. So this is a photo of our mixing pump and mixing apparatus. Was actually heavily borrowed from our USGS partners, and we greatly appreciate their letting us discussing how to actually do this on site. It was quite a trick, but it worked out well. The bottom photo shows a wand system that we actually use. That's basically a pipe, pump pipe wand system, delivery system to the divers underneath. So that's our practicing the wand distribution system in our frame in our parking lot initially. So that's basically our design for getting it out there, getting it mixed on site, and then delivering the product to the divers underwater. We had a lot of monitoring that we agreed to conduct as part of the permit, but also as part of the project to understand the pre, during, and post-treatment conditions. You can see all the water quality parameters there. The key one for us was turbidity, because there's a direct relationship between the concentration of Zequanox and turbidity. So we used the turbidity measure as a surrogate for understanding if we were meeting and hitting our Zequanox concentration levels underneath the containment system. We also included caged mussels as part of the monitoring. Our project partners did the monitoring of those. You can see the cages there. They're very expensive IKEA cages. And that allowed us to monitor mussel density, as well as some of the other parameters that the project partners were taking a look at. We used SONS that you see in the bottom center photo there, and these we're monitoring real-time conditions, especially turbidity, but also some of these other parameters of temperature and conductivity and things like that. So our product concentration and volume, our targets were for turbidity levels were 50 NTUs, which equates to 100 milligrams per liter of Zequanox. So this gives you an idea of we were targeting a 9 to 12 hours for exposure time while we were out there, but we had some weather issues that prevented us from basically required that we get off the water, or we couldn't go out because weather picked up. It was really an unusual August last year weather-wise. Normally it's fairly calm and reliable, but of course when you're doing a study that isn't always the case. So this describes some of the treatment durations, how long we actually had these different exposures within some of our containment areas, and gives you an idea of the range of exposure duration that we had during our study. Even though we had a target in mind, we had as long as 20 hours of exposure and as little as 6 hours. So again, our plans, they actually came together, but uncomfortably so in some cases because they were so late. But it actually worked out in a lot of ways. We planned for a calm lake. We didn't have that, but we were able to spread it over a couple weeks and get all of the plots sampled. And but we did not see any other fish activity besides gobies. We were able to hit our temperature target. We were within our exposure window, generally speaking, and we had to stage it over 2 weeks rather than 3 days. So getting the frames, the covers, and everything set up worked out successfully because we had these great divers helping us with that. They're very experienced in doing these sort of complicated setup things. It worked out really well as far as the layout of the plots themselves, or the quadrants, I guess. But we did need divers to set up this work. So there was that complexity, and diving at 30 feet, they've got a limited amount of time per day when they're allowed to stay down. And then thanks to our great team, we also were able to work with USGS folks to develop a transportable mixing delivery system out to the site. Our pumping plans worked out, fortunately. Our wand delivery system worked out to the divers, and we had great communications throughout with the divers through the audio system to be able to actually watch, along with the video that they included, what was going on down there and adjust accordingly. But we were able to keep a tight seal during the study. We were able to get the concentration levels that we were wanting, and the whole thing actually came together despite all the complexities involved. So in summary, our containment design worked and held. Our delivery system worked. We met our maximum turbidity targets that we were looking for. We didn't quite hit the recommended exposure period, but we're encouraged to learn if that short of a period actually is still effective, because it seems like those recommended targets may be for controlled environments. So we're looking to see how well they work in the open system. We've got a lot of ideas on how to improve it in the future, and we found that the implementation phase, as complicated as it was, actually worked, but it is complicated. So, so how and where you would do this in the future, I think our experience is it would need to be a small site, like about the size of the ones that we implemented for the study, but we were able to actually implement it quite nicely because of all of our planning and the folks that we worked with. So, so I guess overall, we're happy with the implementation. We've got a lot of monitoring that's still going on by the project partners, and I'm sure that folks will learn about the success or not of this particular type of application. And I think with that, got a lot of people to thank. I won't go through all of this, but we really appreciate all the help, and we appreciate the opportunity to work with the Invasive Mussel Collaborative on this really fun study. That's it.
Thanks, Doug. It's Erica again, and I just wanted to reiterate a couple of the next steps and where the collaborative will be going from here. So as Doug mentioned, several of our project partners have been revisiting the site this year to see how the site is faring a year later. So we're looking forward to hearing back from them. And as well, we are putting the finishing touches on the final report, which includes all the work that Doug just went over, as well as the final results and the details of the changes in mussel population and density, as well as the surrounding benthic community, and in addition, the bacterial community, as the botulism issue is of particular interest for the Park Service. So we are pleased with the results and what you will see in those findings in terms of the significant reduction in mussel density, as well as minimal impact to the surrounding communities. So look forward to those results and the final report very soon. From a broader perspective, the Mussel Collaborative is considering this work and other similar opportunities, as well as other research and management priorities. So we have a couple of work groups now that are working to coordinate and prioritize ongoing control method research. And we have a coastal site working group that's looking at identifying other potential sites around the Great Lakes where future pilot projects could be implemented feasibly and hopefully to the benefit of the surrounding community. And like Doug, I have a general acknowledgement slide here. This is a true collaborative effort representing the work and knowledge and expertise of a lot of Mussel Collaborative members and partner agencies. And again, we were funded through the Great Lakes Restoration Initiative with the support of the USGS and other collaborative members. And I will wrap there and turn it back to you, Ceci.
Thanks, Erica and Doug. So that concludes our presentations for today. We will now move on to answering questions and responding to any comments you may have. As a reminder, please use the chat box to submit your question or comment. And with that, we'll start with Diane and Angelique. So the question is, would the use of copper water pipes help control quagga mussels that want to attach to pipes?
So I'll just jump in with that one, that it's not something that we've looked at, but I think there's probably a good chance that it could be effective to some degree. I think it would really depend on, I mean, if you're talking a copper pipe like you use for water intake, I'm not sure that the copper concentration would be high enough, but so I can't really answer that definitively.
Okay, thanks. For Caroline and Doug, regarding your Zequanox field trials, can you just describe how you were able to confirm that the product was applied to the lake as a full strength product, and that it didn't degrade during shipment or through application?
This is Caroline. So I'm looking at the USGS's report right now, and every part of all the Zequanox stuff was actually handled by the manufacturer Marone Bio. So they did store it in cold storage while they were waiting to apply it, and they actually mixed it every morning before application. So they mixed it with the lake water, and they were looking for a certain concentration. So I could send that exact language if you're interested, but to me, it looks like because they did it every day, they were able to get the exact concentration that they wanted.
Yeah, and for our study, we Morone also delivered the product up to the site, and they actually came out on the mixing delivery boat with us the entire time. So they were there the whole time. So we assume that it was fresh product and all of that, because they were actually the one that delivered and helped us implement it on site. So I think that answers the question.
Thank you. Again, for Diane and Angelique, could the increase in chlorophyll A immediately after copper application be from release of the pigment from cells after death?
I think Angelique might address this because she was on site during the treatment, and if I'm not mistaken, that's a possibility, yes, but I'm not mistaken. I think that they also were observing that there was possibly a bloom going on at the time. Is that right, Angelique?
Yeah, there were a couple guesses or hypotheses as to why we saw this. Um, Sometimes around that time, there's a bloom that happens in that area, and so one of the ideas was that potentially that bloom was starting and we did treatment at the same time, and so it was, getting kicked off, and then the treatment cut it back, which is when we saw that decrease 2 weeks later, but it would have been enough time for that initial increase. So I guess we're not sure what it might have been from, but there were several theories that we had.
Thanks. And continuing for Diane and Angelique, is looking at both invasive and native mussel mortality 24 hours after the copper application representative of the true impacts of copper, particularly since bivalves can close their shells and avoid stressors for short periods of time?
Yeah, so part of the reason mortality was measured after the 10 days was to be able to get, look at immediate mortality and also get samples for the copper tissue analysis. And that is true that the mussels will close to avoid noxious chemicals, but this was a 10-day treatment, so whether or not they stayed closed for 10 days is a little bit of a possible stretch. But I should add that that is something that we're looking at in the next study is some delayed mortality. And maybe Angelique can also talk a little bit about the follow-up scuba survey, which was done more than a month after the treatment.
Yeah, so the we had the caged fat muckets and caged adult zebra mussels, and we removed those one day after, and those are the ones that we would have seen that immediate mortality in. The scuba transects were done in September, and were just for the resident adult zebra mussels, and we did see, I guess, great mortality in the test bay of those adult zebra mussels. So that does suggest that there was some sort of delayed response. We don't have that for the native mussels, but that's, as Diane mentioned, something that in the future we would like to do. If we do this type of treatment, we'd like to do like to have some of the cages out and remove those have them in the treatment bay for longer periods of time to sort of track what the response is immediately after treatment, and then going further out after treatment.
And for all presenters, covering both EarthTec and Zequanox, would repeated applications be a better strategy for bivalve control as opposed to just one single application?
This is Doug. I don't know. I guess it depends on what your objectives are. I think some of the interesting things on Good Harbor Reef are after the initial treatment, do the gobies actually come in and sort of keep the area unoccupied or less occupied by recolonization than going back out and having to retreat. So for a natural system, I think you probably want to try to not retreat too many times, at least my impression. but that might be different in sort of an industrial setting or some other man-made setting.
And this is Erica. I'll jump in and add, this kind of question has come up with the Mussel Collaborative and is reflected in some of the strategy objectives that we've put together. So one of the questions the collaborative is considering or would like to see addressed is, how long do you have to implement, sustain, repeat treatments? Are they needed in order to achieve a level of suppression that's needed to generate the benefits you're looking for? So that's one of the questions that's come up within the collaborative. With this particular project, unfortunately, our focus was more on feasibility of treatment in a coastal environment and what the potential benefits could be. But that issue of, are continued repeated treatments needed to keep an area free of mussels, or at least suppressed to a level where those benefits are realized, is a question that's on the collaborative mind and one that we are looking at addressing.
Yeah, this is Diane, and I would just add, as far as the copper treatments, that question has also come up before. Instead of a 10-day application of copper, could you just do it for a couple of days at different periods throughout the season when veligers are being produced? And that is something, that deserves to be looked at. But I think that, again, the trade-off is if you're looking at repeated applications and there's harm to non-targets, you're repeating that harm to them every time you do the application. So it's, as you mentioned, Erica, those are questions we would definitely like to address.
All right, for Erica and Doug, as monitoring continues at Good Harbor Bay, will you be monitoring recolonization rates?
So as I mentioned, we were able to do some monitoring this year in conjunction with the Park Service and University of Wisconsin-Milwaukee, who is also continuing to monitor the nearby sites where they conducted their manual removal effort in 2016. So we were able to go back out and do that monitoring this year. So we'll have an idea of how the site has changed and what the status of mussels are a year later. Future years monitoring will be dependent on availability of resources. We certainly hope to be able to continue that in some fashion, but as I mentioned, we're dependent on the availability of future resources to do that work. But it is, again, something that the Mussel Collaborative has recognized. As you think about long-term management strategies and selecting your sites where you're going to do those, what is the potential for recolonization and what is the rate, to help answer that question of, how long do you need to sustain management activities or suppression to achieve those benefits?
Thanks. Back to Diane and Angelique. How did mortality of native mussels compare to the mortality of invasive mussels?
You want to take that one, Angelique?
Yes, it helps if I can find my unmute button. Sorry about that. So we had in our cages, I'm just pulling this up to make sure I'm looking at the right thing. In our caged animals that we removed one day after treatment, we had very good survival of the fat muckets. I think almost all of them survived. And we had slightly lower survival of the zebra mussels. I think nearly all of them, again, survived in the control bay, and somewhere in the ballpark of about 65% on average survived in the test bay. So it looks like we had slightly higher mortality amongst those caged animals. And then again, as I said before, we did have very high mortality among the resident adults that we tracked in our scuba transects. But unfortunately, we don't have that information yet on how the native mussels did in the long term. So I can only
I'll
hope
just,
that.
yeah, go ahead. I was just going to add that the mussels that were used were not new juveniles, but they were subadults. They were pretty young. So similar in size actually to the adult zebra mussels. And again, it would have been nice to have the delayed post-treatment sample to see whether or not there was any delayed mortality.
And continuing with Diane and Angelique, regarding St. Albans Bay, did you have any thoughts on the cause of mortality among the benthic invertebrates?
Oh, that's a good question. I don't know. Diane, do you have thoughts on that?
Well, I will say that it's, you know, these invertebrate populations are obviously fluctuating pretty on a pretty short cycle. So it's really kind of hard to tease apart the effects on the treatment versus what was just some normal seasonal change in the population. But having said that, I mean, there's pretty established toxicity data for copper, you know, to various benthic invertebrates. So, you know, as quickly as 1 day post-treatment, but you have to remember that they had been exposed to it for 10 days before that 1 day post-treatment sample was taken. So I think it does represent that there is likely some non-target toxicity to those benthic invertebrates. And as Angelique mentioned, we have the species composition and abundance data that will be analyzed as part of this study. It just wasn't presented today. So that will give us a better idea of whether or not the treatment is affecting species composition too.
And then I think we've got one question left that I think could be addressed by any of our presenters. Is there knowledge of, you know, the actual biological process by which the Pseudomonas in Zequanox kills the invasive mussels? And if so, would there possibly be an opportunity to genetically engineer some plankton species to carry that bacteria to mussels in a live format?
Nobody wants to jump in on that one. I will just mention we do have some research going on in our center. Chris Merkus is our geneticist, and he's working on an RNAi tool, so not a chemical tool, but a molecular tool to incorporate into probably bacteria for delivering a lethal molecular tool that is very selective for dry cenids. So that's how I can answer that one.
Yeah, this is Erica. I don't have a specific answer other than to say this would be an interesting question to bring back to our research work group and the Mussel Collaborative, which Diane also co-chairs. So we can consider this as we look at ongoing research and identifying priorities as a potential question to look at.
All right, well, we are right at the top of the hour, so that's all the time we have for questions right now. Thank you to all of our presenters and participants in the webinar today. As a reminder, this webinar recording will be posted on the Invasive Mussel Collaborative website within the next couple of days. And if you have any additional follow-up questions or comments, please feel free to contact us at the Great Lakes Commission, and we will get you in touch with our presenters from today. Thank you.
Thank you.
Thank you.